Today I will be dealing with another ingestive function of the hypothalamus. In the last class, I had taken the feeding regulation by the hypothalamus. That is one of the ingestive function. And here in this lecture, I would be discussing on the water regulation, especially in connection with the thirst. You see in the picture there on the right hand side, a thirsty man trying to drink lots of water. You see that urn that is full and he is trying to empty everything there. That means he is thirsty. So we consider this thirst mechanism in this lecture. Of course, thirst mechanism is one of the component of the regulation of the water intake. Now, just continuing with the previous, we have covered these first three topics. Today I will be concentrating on the water regulation by the thirst and post-respiratory. Okay, so now in this I have mentioned the daily water balance, average water balance by an adult. You know, we consume nearly 2 liters or 2.2 liters of water through food and drink every day. And we get about 300 milliliters of water coming from the metabolism of the various food products. Especially, you say that carbohydrate, when it is burnt, it is the glucose, when it is burnt, it gives the 6 moles of carbon dioxide and 6 moles of water. So like that, the water is coming there by the metabolism. So total daily water gain, either by the ingestion or by the metabolism, is around 2.5 liters or 2500 ml. Of course, we also lose water, or there is a water loss every day. One thing is by insensible water loss via skin, because we all perspire without our knowledge, insensible water loss through perspiration. That is one component. That may be amounting up to 500 to 700 milliliters. And we always expire because you think that 15 breaths a minute, and you calculate in 24 hours how many breaths we take. And each breath will expire out some amount of water vapor which is present in the lungs or in the body. So that will also lose around 200 milliliters of water. Total loss by the insensible water loss by the skin and lungs is about 900 milliliter. I have marked it red because we cannot avoid this. It is going to happen. Okay, here also I have made it red here, metabolism, because this is the product, because it cannot be changed. Then we lose water by the kidney through the urine, and it is about 1500 ml or 1.5 liters per day. This is, I'm talking about the average. So now we have in an extreme conditions what is called an obligatory volume of urine. This volume of urine, which is necessary to keep all the constituents of the urine in solution, that is about 500 ml. 500 ml is the minimum volume of urine which is required to keep all the solutes in solution. So this 500 ml is obligatory. This much is required. So that means this 1500 ml can be compromised to 500 ml. That means 1000 milliliter is extra that can be regulated. Then we have water going through the stools or feces. So that is not much as compared to the kidney. It is about only 100 milliliters. So this is fixed. So that means the insensible loss is fixed. The loss through the feces is fixed. The only thing is coming here is 1500 ml. The urine volume can be altered. That can be variable. So the total amount is 2500 ml. You have a perfect balance sheet there. It is 2500 ml intake, 25 ml output or whatever loss. So that is totally perfectly we are balanced, and we don't have any excess of water. What happens if you have excess of water? What happens if you do not have enough water? That means we keep losing water. See, it may happen in case say for example the diabetes insipidus, the water is lost. So what happens? So those are the things we have to consider today. So considering the water balance, now we just move on. The water balance is regulated by the number of mechanisms. I have listed the number of mechanisms. One, by the ingestive behavior, by the osmolar activity, the osmolality of the extracellular fluid and the plasma. This osmolarity depends upon the ionic components, ionic things, that is electrolytes, or non-ionic, that is a non-electrolytic. That means a glucose, mannitol, or urea. They are non-electrolyte molecules. They will contribute to the osmotic activity. So I will not consider here the proteins. Proteins have a charge. They have a charge, so they may be having either a positive or a negative charge depending upon the pH of the blood. So, but the osmolar activity is dependent upon two components here, ionic and non-ionic components. That also regulates the water balance. Third component here, the water balance is regulated by number of hormones. I have listed the number of hormones here. Antidiuretic hormone, aldosterone. Antidiuretic hormone is purely water regulation. Aldosterone is a sodium regulation. Water comes as a second. Then the other one is opposite of the aldosterone, the atrial natriuretic peptide, or a brain-derived natriuretic peptide. So these are hormones which eliminates sodium from the kidney, and that sodium goes along with the water. So that is it is opposite of the aldosterone here. Now we have another hormone, the angiotensin II. So this angiotensin II regulates, one, the vascular smooth muscle activity. Two, it stimulates the centers in the brain. Three, it stimulates the secretion of aldosterone from the suprarenal cortex or adrenal cortex. Now, the fifth hormone I am mentioning here is relaxin. Relaxin is a hormone which is secreted from the decidua and also from the endometrium. And this relaxes the smooth muscle of the myometrium. But this relaxin produces increased water intake. So that means during pregnancy, the lady is taking more water, but she does not have the hypotonicity. So these are hormones which are regulating. Besides these things, we have cortisol, that is a corticosteroid. That is because that is having a mineral corticoid activity. We have estrogen, so they also try to hold on some amount of water, especially certain parts of the target organs. Even if you are thinking about, you are very meticulous about the things. That means the prolactin. So that may also hold water. That holds water in the breast. So like that, we have other hormones, they may regulate, but these are main primary hormones. Now, the neural regulation, The neural components that regulate the water balance is a cortical, somatosensory. That means we have a desire to drink water, so that means that will activate the sensory component. Once the sensory component is activated, we have these the behaviors associated, the prefrontal and frontal areas are activated, then motor cortex jumps in, and we try to drink water. That is one, the neural component. Besides that, other factors, maybe I will come back later. So then second neural element which is participating in the water regulation is the limbic system. That is a cingulate gyrus, that is the behavior, water behavior. So that means whenever you go to somebody else's house and they will offer you a glass of water, though you may not be thirsty, you will drink it. That's a social behavior. And the behavioral aspect, the amygdala. So that means that also try to ingest water or try not to ingest water. So that means we inhibit or it may facilitate the water intake. The fornix, the area in the limbic system, that would also increase the water intake. The hippocampus, that is associated with the memory. So now these are all. So then mesolimbic system. Mesolimbic system is a reward center, reward and punishment center. These reward and punishment centers are associated with the psychological elements or psychological components, so that they will try to influence the water intake. So that means psychologically you may be drinking water. Say for example, what I am telling, you are trying to get out of the house. When you are trying to get out of the house, you feel that, okay, whether you may get water or you may not get water. So you go there and you open the fridge and then you have a water, or in the hostel, if you are there, you have a bottle and you just try to drink water. So that means we are mesolimbic system, because you anticipate certain things. You anticipate certain things that you may not get to drink a water for some time. So now, then hypothalamic centers that I will separate. There are several hypothalamic nuclei which regulate the water regulation. Then we have a circumventricular area in the brain. This is a very peculiar area. The circumventricular area is a peculiar area, so where the blood-brain barrier is not intact. So that means it is the place where the blood-brain barrier is broken, or it is not continuous. It's discontinuous. So that it allows the communication between the external part to the brain areas. So this is circumventricular areas that I will talk about. So then we have a water regulating centers. These are the centers which either produce the ingestive behavior or will activate or sense the ingestive behavior. That is the NTS, parabrachial nucleus. So these are. So then this NTS and NTS is communicated with the baroreflexes. Okay, these are the neural elements. So the water balance is regulated by four things. Ingestive component. Ingestive component is thirst, drinking, eating. Because while eating food also we drink water. We drink water. I'm sorry. So now, these four components: ingestive, osmolar, hormonal, neural. Now we move on to the next. What is thirst? Because here, if you go back, thirst was the main thing. So now we want to know, or today I will be mostly focusing on this component of the water balance. Others also I will tell, but mostly I'm concentrating on the thirst. To define thirst, it is a subjective perception that provides desire for human beings or animals to drink water or ingest water. I repeat again, thirst is a subjective perception that provides the urge for humans and animals to drink fluids. It is a component of the regulatory mechanism that maintains body fluid homeostasis, ultimately essential for the survival. So that means it is a essential component of the homeostatic mechanism, and it is an urge to drink fluid. The urge to drink or urge to ingest fluid arises from number of factors that may be habitual, that may be cultural. Habitual means we are habituated to drink water. So whenever we come from the hostel, we drink water, or we go to the hostel, we drink water, or wherever we see the water resource, maybe we may drink water. So this is habituated. And cultural means, I said, whenever we visit somebody, they may offer you some drinks, beverages, and the water, plain water, or anything, or cold drinks. And psychogenic drives, so that means that psychologically you are stressed up, you may start drinking more water. Or psychologically you are going to a distant place and you are trying to compensate for that as an anticipatory mechanism. Besides these, there are conditioning components associated with the water drinking. Conditioning components in the sense, just like a conditioning reflexes. Suppose if somebody is habituated for drinking tea somewhere around 4 p.m. So then at 4 p.m. he needs to drink that tea even there is no water imbalance or a fluid imbalance. So these are conditioning part. So these regulatory response which reduce the fluid content of various, okay, they are coming from the reductions in the fluid content of the various compartments of the body, or resulting from the hypertonicity of the extracellular fluid, or increase in the circulating concentration of some dipsogenic hormones. I have already mentioned those dipsogenic hormones. I again say these dipsogenic hormones may be ADH, maybe angiotensin 2, maybe aldosterone. So these things you may consider to remember. Okay, next we will move on. This again I will come back regarding thirst. It is an unpleasant sensation of desire to drink water, and it is associated with the dryness of the mouth and throat. So that means even this unpleasant sensation, because you suddenly feel that you need to have water, so you have a sensation to need drink water. So mouth is dry and throat is dry, and you try to. Now, so there are certain experiments done. So the people thought that it is because of the dryness of the mouth that results in the that unpleasant sensation. So to avoid that dryness of the mouth, they applied local anesthetic in the mouth, that is mouth and the throat. So when they applied local anesthetic, the thirst, the unpleasant sensation did not disappear. So that means it is not just because of the dryness. It is something inside it's happening. And another experiment was performed. In this experiment, what they did is in dogs, they have an esophageal fistula, and this esophageal fistula, what they did, they have the esophagus connected to they exteriorized the esophagus. They allowed the animal to drink water, and whatever the water the animal drank, so that was eliminated through that fistula outside. So that means, is it a psychological thing that we are drinking water and wetting the throat and mouth? Is that going to help? No. Still, then these animals felt thirsty. So that means it is not the local factor happening. Okay, I may agree. Local factors may be working to an extent, so that whenever you are thirsty, you have something to wet your saliva, to wet your tongues and the mouth, and trying to keep it these things. But the inner feeling does not abolish. So, but this urge to drink water disappears the moment we drink water. So that means you just think that when the water you have drank, it reaches the stomach, suddenly it disappears. maybe whatever route, maybe I'm talking about any route, here I mean to say the intravenous route, or you can also by oral route, or you can also supplement the water, or give water through the enema, that means a rectal route. So any route if you give, the the satisfaction is there. That means that the urge to drink water disappears. Now, it was thought that the dorsal and ventral hypothalamic areas participate in it. And subsequently, the people thought that the paraventricular and the periventricular areas are involved. So that is what the previous knowledge. Now, going further, even in the earlier some books, you may have certain books, they may be mentioning about these things. The paraventricular nucleus has been identified as one of the center, because local injection of the hypertonic saline in this area produced the copious drinking behavior in the animals. So that means they drank too much of water. In rats and cats, they performed. So then that is one, that means they identified with the paraventricular nucleus. Now, the stimulation of the Wick-D Agire. Wick-D Agire is a mammillothalamic tract. Mammillothalamic tract, it is also known as a Wick-D Agire. So when they stimulated the mammillothalamic tract, that forms the part of the peptide circuit, you remember. Or when you stimulate the area of the fornix, fornix is again coming from the dentate gyrus, and it is going to the septal nucleus. So that means the fornix. Fornix is the area, so that forms the dorsal hypothalamus. Okay, when they electrically stimulated it, the animals exhibited polydipsia. So that means they are concerned with the regulation of the thirst mechanism. Electrolytic lesions of these, the mammillothalamic tract or Wick-D Agire or the fornix, they produced the loss of drinking behavior, hypodipsia. So that means these are the two, there are two areas now I'm talking about: paraventricular nucleus, mammillary body, and the fornix, which are part of the dorsal hypothalamus. They are involved in the drinking behavior. However, the people, when they try to examine these old experiments, the lesions produced or the stimulation produced were extensive or broad, so that we cannot pinpoint, because if you are trying to look at only fornix area, it is stimulating the surrounding area. What is that hypothalamus? Hypothalamus is a small area, and you are trying to stimulate, maybe you cannot isolate the neighboring areas. It's very difficult, extremely difficult. So now we cannot pinpoint the particular areas. Now we know precisely these areas nowadays. Maybe we may go it further. So these are the circumventricular organs. So I mentioned about this. These are the areas in the brain which are having some amount of the deficiency of the blood-brain barrier. You start from the OVLT, organum vasculosum lamina terminalis. Organum vasculosum lamina terminalis. This is the area here in the suprachiasmatic region. This is one of the area wherein it is freely accessible to the extracellular fluid changes or the blood changes. Now we have the postpituitary, this component, the median eminence. Then comes here area postrema in the medulla. So then we have here the pineal gland or pineal gland. Then we have a subcommissural organ and subfornical organ. So that means 1, 2, 3, 4, 5, 6 or 7 of these are there. So if I repeat again, pineal gland, the subcommissural organ, subfornical organ, OVLT, organum vasculosum lamina terminalis, OVLT, the posterior pituitary, the median eminence, area postrema are all belong to the circumventricular parts. So of these circumventricular parts, we are these OVLT, the subfornical organ, and the posterior pituitary are the main one for the water regulation. Let us consider it further. Now, whatever with the current knowledge, what we know, what are the areas in the hypothalamus which regulate the water? So now, as I have shown in the previous graph, the OVLT is one, that is one, and subfornical organ is the second. So these two are the prime areas of the hypothalamus that regulate the drinking behavior. So actually in the OVLT we have those osmoreceptors. Okay, subfornical organ which regulates these osmoreceptors. Then after that, because they are with the advantage that they are in vicinity with the extracellular fluid, whatever happening in the extracellular fluid can be easily detected in these organs because of that circumventricular, belong to the circumventricular organ. Now, then in the other things, the paraventricular nucleus, the medial preoptic nucleus, the supraoptic nucleus, the periventricular area, that means around the ventricles, and we have a lateral hypothalamic area. All these things participate in the thirst area in the hypothalamus. But the red ones I have made here, they are prime. Then comes we have the paraventricular and the preoptic area. So I almost have written in the order of their ranking in regulation of the thirst mechanism. Now, brain areas involved in the drinking, because here in this slide I have shown only the hypothalamic areas that regulate the thirst. Now here I show the entire brain areas, they regulate the drinking behavior. So now entire brain areas, I have already mentioned about the hypothalamic areas. But these hypothalamic areas now are associated with the limbic areas like a cingulate cortex, the central nucleus of amygdala, the hippocampus, the prefrontal area. This is prefrontal area. This is cingulate cortex. This is cingulate cortex here. So then prefrontal area. This is prefrontal area. Then mesolimbic area somewhere here. So then cortical and subcortical areas involved in the ingestive behavior. These are cortical areas which are involved in the ingestive behavior. So what is that ingestive behavior? That is drinking of water. Drinking of water, you have to do certain set of reflexes. You have to elicit certain motor activities. For these motor activities, the motor cortex is involved. The sensory cortex is involved. So those cortical and subcortical areas which are involved in the ingestive behavior, because these are automated reflexive activities of the animal or the man to drink water. Suppose even if I have to drink my own saliva, so I did swallow my saliva, and when I swallow this saliva, I initiated certain, so it can be started voluntarily. So that means what? The motor cortex is involved. So later on it goes on. So that means the cortical and subcortical areas involved in these voluntary act of the ingestive behavior. That's one component. Then the cortical and subcortical areas are especially the cortical areas involved in the conditioning of these reflexes, because we are having a conditioning reflexes for the drinking behavior. So now this conditioning component also, these are the areas. Then we have the above the medulla and the pontine, medullopontial junction, this parabrachial nucleus, lateral parabrachial nucleus involves in the water intake, because that communicates with the hypothalamus and the cingulate cortex and also amygdala. Then we have the nucleus tractus solitarius. that receives the vagal inputs and that receives the inputs from the glossopharyngeal nerve, regulates the blood pressure, the visceral component. So this nucleus tractus solitarius is also involved. Then we have Raphe nucleus. Raphe nucleus is involved in the drinking, and they are suppressing, because this is where we have the vomiting center is located. So these are all the areas which are participating in their drinking. I repeat, the hypothalamic areas. Those hypothalamic areas are primarily, if I were to name, OVLT, SFO, that is a subfornical organ, then a paraventricular nucleus, medial preoptic nucleus, then lateral hypothalamus, then supraoptic nucleus, like that. Then we have the limbic components, the cingulate, central amygdala, hippocampus. Then we have these the higher functions or social type, the prefrontal area, then reward centers, the mesolimbic area. So then conditioning centers and other pattern generators of the cortical and subcortical areas, then the medullary areas which are receiving the inputs. This is all concerned with the drinking. Now, so if this graph I had shown in my lecture in the ADH secretion. Just I'm trying to bring back this slide, trying to explain, because it is one of those component. This is an osmoreceptor here. Now these osmoreceptors are present in the OVLT, and these osmoreceptors are activated by the change in the plasma osmolality. The change in the plasma osmolality is not, you need not to have the extensive change in the plasma osmolality. Even a small change in the plasma osmolality is detected by the osmoreceptors. And these osmoreceptors may be because of the increased osmolality, either by electrolytic medium or by the non-electrolytic. Say, for example, I want to give one example. In a diabetes mellitus, the blood glucose level increases. The increase in the blood glucose level, so that increases the osmolality of the plasma. And this osmolality of the plasma directly increases the osmolality and stimulates the osmoreceptors. So now, if I go back with the diabetes mellitus, polyuria, polydipsia, and polyphagia, these are the three things we teach. So this polydipsia is because of the increased osmolality that would stimulate the osmoreceptors. Then I have another point I have written here, indirect. So that means in a person with a loss of water, loss of only the water component, so electrolytes remains. So then there is a relative increase in the electrolytes and other osmotically active molecules. So that will produce the indirect, whatever may be the reason. So that will stimulate the osmoreceptors. Osmoreceptors in turn the paraventricular nucleus and the supraoptic nucleus, they will try to manage with the EDH. And that they will stimulate the ADH secretion. ADH try to reabsorb water and try to compensate. Mind you here, so this is regulation of the water I'm talking about. Then at the same time, what they do, this would stimulate the NTS. Or this would stimulate the other component, the sensory component, wherein the ingestive, he feels the urge to drink water. So that means the medullary sensory, somatosensory components are activated. So urge to drink water, he will have or begins. Now, the second component is because of the increased plasma osmolality, or if there is a decreased plasma volume, there is a blood loss. The person with a blood loss, have a decreased blood pressure. Decreased blood pressure do not activate the baroreceptors here. Do not activate the baroreceptors. So if they are not activated, so that means they will not send the inputs, the inhibitory inputs to here, and the NTS have an inhibitory input to the osmoreceptor. So that means the no inhibition is a facilitation that will activate the osmoreceptors and the paraventricular nucleus. That is how this is happening. Here I have mentioned decreased plasma volume, decreased barodischarge, decreased excitation of NTS, decreased inhibition of hypothalamus, and the water secretion. At the same time, this increases the sympathetic activity. Maybe I will come back. The sympathetic activity have a dryness of mouth and the drinking behavior. Now, so I will move it further. This one is the overall the regulation of the thirst. So I will take time here. So now these are the osmoreceptors in the hypothalamus. This is the thirst center. Thirst center for me are the OVLT and SFO. That is the subfornical organ and the OVLT. Then osmoreceptors may be there in the OVLT and these things, and this medial preoptic nucleus and the paraventricular nucleus here in the regulating these. So anyway, I have made it as a hypothalamus there. Now let us start with it. There is insufficient water in the body. That is the initial part. Now, if there, because of the insufficient water in the body, what happens? The plasma osmolality increases. You can just see that water is lost. So relative component I was trying to talk about, the osmolality increases. So increase in the osmolarity, what happens? It stimulates the osmoreceptors present in the OVLT. And once it stimulates the osmoreceptors, it will activate the third center. And once it activates the third center, so third centers will stimulate the somatosensory or all other part in the medulla, that reflexive component to the cortex and the medulla, cortical and subcortical areas, to drink water. So that means you try to go and find water and drink. So that means the entire behavior is associated. So to drink, there is an increase in drinking of water. It has started with the increased osmolality. Now water intake is activated. So now, so then comes, this is okay, that is fine. Now what is happening here? Maybe if I come, the increased osmolality produces the dryness of the cellular dryness, dryness in the mouth and the throat. Now the dryness in the mouth and throat stimulate the osmoreceptors. How they stimulate? They stimulate osmoreceptors because the glossopharyngeal nerve or the nerve supplying the pharynx, they are carried going to the medulla, and they will activate the osmoreceptors. So that means through the parabrachial nucleus. That is one component. The second component, we have these third centers are directly activated, that would produce the water intake. Then we come back with the second component here. The insufficient water produces the decreased blood volume or plasma volume. So decreased plasma volume will activate the osmoreceptors by the decreased blood pressure. The decreased blood pressure that results in the baroreceptor activity, decreased baroreceptor activation. Decreased baroreceptor activation through NTS, we will activate the chemoreceptors. So that means it is a osmoreceptor. Through the osmoreceptors are activated because of the decreased blood pressure and decreased baro discharge. Okay, so now this will increase the sympathetic activity. I mentioned here sympathetic increases the third center. This will increase the sympathetic activity. Sympathetic activity produces the dryness of the mouth. So whenever you are nervous, whenever you are coming to the examination, or you are having the anxiety or those things, your mouth will dry because of the sympathetic activity. So now this sympathetic activity will also be increased because of the decreased blood pressure. Okay, so now what happens further? You go back to the kidney. In the kidney, what happens? Renin secretion increases. The renin secretion in turn increases the production of the angiotensin II. So you think that renin-angiotensin mechanism. So that means angiotensin II is produced in the lungs, mainly in the lungs, because angiotensin converting enzyme is greatly expressed or greatly present in the lungs. So angiotensin II is formed. Now what this angiotensin II? This angiotensin II produces the vesicular the smooth muscle constriction and try to increase the blood structure. That is one component. So, but this angiotensin 2 is not only a neurotransmitter, it is increased in the entire body. It is acting like a hormone, and it would stimulate the third center. Angiotensin 2 is stimulate the third center. In the third center, the angiotensin 2 is a transmitter. So that means it will enhance the third center, and that will increase the drinking behavior. Suppose if I inject micro iontophoretically, that means with a very specifically inject at this third center, so it will increase the water intake or dipsogenic behavior in the animals. Okay, so now coming back with what this angiotensin 2 does. The angiotensin 2 increases the aldosterone secretion. This aldosterone in turn reabsorb water from the distal nephron, mostly the distal nephron. So this what sodium is reabsorbed. Now the sodium comes along with the water, so that means that will try to increase the extracellular fluid volume, and this increased extracellular fluid volume trying to balance the decreased blood volume. So that means that would compensate for the decreased blood volume. So now one aspect I said, the decrease in blood pressure activates sympathetic nervous system, that would try to increase the blood pressure, and it will that will increase the angiotensin 2, that further increases the blood pressure. We have already the sympathetic and angiotensin 2 activation. This is one of the component we have seen. Now coming back here, the increased water intake, what it does, it increases the plasma water content. If the water content is increased, so whatever increased plasma osmolality, now it is balanced. So that means that is stabilized. So now once that is stabilized, the dryness of the mouth and all other cellular dehydration will abolished, and suddenly you feel good. So that is one component. Now, if you are looking at the supraoptic nucleus and the paraventricular nucleus, I am taking them as separate, that regulate the ADH secretion through the posterior pituitary. So now they would activate the posterior pituitary to secrete the ADH, and this ADH, what it does, it increases the water reabsorption. That means increases the water activity. So that is another component that is coming up. So that is coming up here. That is coming up here, so that increases the water intake. Now I have two hormones here, the atrial natriuretic peptide, because if there is a increased atrial structure, that would try to suppress the third center, because there is too much of water. Say for example, I'm talking about not insufficient, the greater amount of water. In that case, the atrial natriuretic peptide try to that inhibit the third center. So this is one component. I have mentioned about sympathetics and relaxin, that would excite the third center so that water intake is enhanced. So now sympathetics also produce the dryness of the mouth. So this is overall a broad view of the how the third centers in the hypothalamus are activated, and how these osmoreceptors and third centers are interrelated. Going little more in detail, and this is one of those thing taken from the news in Physiological Sciences. Okay, I just quickly rush through these things. Here is the OVLT. So here OVLT is a regulator of the osmolarity. Okay, so now as the osmolarity increases, the OVLT detects, and once the OVLT detects, so this would be passed on to the medial preoptic nucleus. And now this medial preoptic nucleus activate the higher integrative centers. Higher integrative centers in the sense, here the sensory cortex, the motor cortex, the limbic cortex, the cingulate gyrus, all those integrative centers, they are activated. Plus the brainstem integrative centers for drinking behavior. All those things are activated, and that would initiate the motor pattern generation. That means drinking behavior, search for the water source, and a drinking behavior, and drink water. That is the uh, quenching of the thirst, acquisition, and ingestion water. So that means these higher integrative centers is for the acquisition or ingestion of water. So this is one component. I have said OVLT, it is going to activate the medial preoptic nucleus. Okay, now come back here. So what is happening? This also activates the subfornical organ. Now, this subfornical organ that would activate the medial preoptic nucleus, and this activation, that means it receives the neurons here. These neurons are angiotensinergic neurons. These are angiotensinergic neurons. They stimulate the medial preoptic nucleus, and that would activate again this acquisition and the ingestion of water. So now, supraoptic subfornical organ also activate the higher integrative centers, as I have said, the cerebral cortex, the cingulate cortex, or the limbic cortex, whatever. So they will just increase. Okay. So this in turn, the subfornical organ in turn is activated by angiotensin 2. Because what is happening in the renal JG cells, juxtaglomerular cells, because of the baroreceptors, the decreased fall of pressure activates these renal JG cells, or the chemical sodium level that would activate the renin release, and renin activates the formation of converts the angiotensin to angiotensin 1. Now angiotensin 1 is converted into angiotensin 2 in the systemic circulation, mainly in the lungs. Okay, that forms angiotensin 2, and in the SFO, that is in the subfornical organ, the 81 receptors are expressed. The angiotensin 1 receptors are expressed. They will enhance the activation of the medial preoptic nucleus and the ingestive behavior. Now here the relaxin excites the ingestive behavior through the SFO, and the atrial natriuretic peptide suppresses or inhibits the subfornical organ for the ingestive behavior. This is what happens in the hypothalamus. These are the hypothalamic centers. Now coming back, so here, somewhere here only, we have the paraventricular nucleus also, that is been that is this, because the medial preoptic nucleus and paraventricular nucleus, they are going hand in hand here. Now coming back here, the other components, renal baroreceptors, they will be making angiotensin 2 and activating. Now arterial baroreceptors, because of the fall of the pressure increases the activates or have a inverse relation here. The area post rama, that is one of those circumventricular organ, and the NTS, they will stimulate, or they will try to stimulate or increase the input to this SFO and the even to the OVLT. So that is not shown there. It's not shown there, and they will inhibit the lateral parabrachial nucleus. Now, lateral parabrachial nucleus excites the medial preoptic nucleus. Now, this NTS gives the inhibitory input to the lateral parabrachial nucleus, which in turn will activate this thing. Now, decrease in the NTS activity, the inhibition is lost. There is a withdrawal of the lateral parabrachial nucleus, so that ingestive behavior is enhanced. That is one component here, coming with the arterial baroreceptors. Now we have the cardiopulmonary receptors. The cardiopulmonary receptors, they detect the stretch or atrial receptors. These cardiopulmonary receptors, they will detect the blood volume, and when there is a decreased blood volume, they will also activate the NTS. That again happens in the same as that of the arterial baroreceptors, in the same pattern. That means inhibitory activity that suppresses the NTS activity, suppression of the suppression, the increased activity of the medial preoptic nucleus or paraventricular nucleus or supraoptic nucleus. Now coming back with the visceral osmoreceptors. We have the osmoreceptors here. These are central osmoreceptors. We have in the viscera, especially the liver and the mesentery, and they have a certain receptor, osmoreceptors. These visceral osmoreceptors, they will detect the osmolality, and they will try to again activate the area, the NTS area, or in the area of the medulla, that would enhance the or activate the medial preoptic nucleus to bring about the ingestive behavior. So now I have explained the visceral osmoreceptors, the cardiopulmonary receptors detecting the volume, the arterial receptors detecting the pressure, and the renal receptors producing the renin-angiotensin axis, and all those things produce the ingestive behavior. Okay, now coming back here, now what is happening? I talked about only that ingestive behavior. If we are trying to look into the water balance, in the water balance, we try to look into as such the hypothalamus, osmoreceptors, the paraventricular nucleus, ADH trying to reabsorb water from the distal convoluted tubule and the collecting duct. So reabsorb water from here, and once they reabsorb water from here, that would increase the plasma volume, and that would try to compensate for the whatever the decreased plasma osmolality. So these are the various variables which will either activate or suppress. So caffeine, alcohol, norepinephrine, atrial nitroglyceride, they will decrease the whatever the input coming from the supraoptic and paraventricular nucleus, and exercise, stress, pain, nausea, hypoglycemia, angiotensin to enhance the ADH secretion from the hypothalamo-hypophyseal neurons. Okay, so now we move on to the next. To summarize, what is the water intake? What we were trying to look at. In the summary, I have just put it here. So this water intake is balanced by the ionic, osmotic, humoral, neural, and cardiorenal parameters. Ionic parameters which detect the thirst mechanism, either sodium, calcium, and to some extent chloride. So these things, they will stimulate the water regulation because of the hyperosmolality or hypoosmolality, whatever, through the osmoreceptors. Now, again, the osmotic I'm talking about here, it is a non-ionic osmotic activity. Say, for example, I gave you an example of the hyperglycemia and producing the polydipsia. So that means increased water intake in a diabetes mellitus patients. So that is the osmotic activity. That will be through the OVLT and the subfollicle organ. Then humoral, I mentioned a number of hormones which are regulating these things. One of those hormones is angiotensin 2 first for the water intake. Second is the antidiuretic hormone. Third is the aldosterone. Fourth and fifth are the relaxin and the atrial nitroglyceride, and you have other hormones which regulate the water intake. The neural mechanisms. So I have mentioned here the hypothalamic areas. So these are the main primary areas of the hypothalamus: OVLT, SFO, PVM, medial preoptic nucleus and supraoptic nucleus, and periventricular area and lateral hypothalamic area. These all participate, but these first four are the important one, or first 4 or 5 are the important one which regulate the water intake. The cardiorenal, that is happening through the baroreceptor and angiotensinergic mechanism, increasing either sympathetic activity or angiotensin 2 activity. So this is how the whole thing, each are interdependent, they will try to increase the water intake. Okay. In comparison to the food regulation, in the food regulation, what happens? We have the feeding center and we have the satiety center. Now here we do not have the satiety center, so that means which can stop. So there is no satiation of thirst center. So now what happens? The minute the fluid balance is maintained, we feel satisfied. We are satisfied. So that means it is only one center, or these multiple centers, they will be either activated or not activated. So that phenomena, there is no second center or a dual centers which will be doing the satiation. So there is no satiety center for the water intake. Now, just summarizing, this is subfornical organ, just trying to give you how those interneurons are connected. So this is the subfornical organ. From the subfornical organ, we have the medial preoptic nucleus here in the hypothalamus. Okay, so now from the subfornical, because if you are looking at the subfornical organ becomes the major cluster here, it carries the cluster here, and this has a connection with the OVLT. Okay, then this has a connection with the supraoptic nucleus. This has a connection with the paraventricular nucleus. So these two have a connection with the postpituitary. So here is the brainstem area. Here are the drinking and the osmoregulation, and these are OVLT area, and these eating, other centers of the hypothalamus. And besides that, what is missing here in this is the subcortical and the limbic and areas which try to influence this. That is missing here, and other hormonal factors that is missing here. But if I am only concentrating on the hypothalamus, if somebody asks me what are those centers, yes, if by looking at these centers, you can just see that the main cluster is here in the SFO and OVLT. So these are OVLT detects, they have the osmoreceptors, SFO organizes, and these all have a bidirectional. Though it is unidirectional here, they have a bidirectional synaptic contact that would produce the overall activity. So this is in brief, or I summarized in this figure, the entire the thirst mechanism. So what is missing in this thirst, and how the ingestive behavior, I did not go into detail, because the ingestive behavior is the activation of the swallowing and searching for the water intake. These are behavior components. They require a lot of other areas of the brain to be coordinated to achieve that. Now, so these are the keywords for this lecture: thirst, osmoreceptor, circumventricular organ, organum vasculosa lamina terminalis, subfornical organ, because you don't forget them, dipsogenic factors or dipsogenic hormones, the brain areas involved in the drinking behavior. So these are some of those questions you may receive. Okay, so now hope I have made it thirst. I have made it in the sense as an urge to drink water. This is a behavior, and it is sometimes very much required, and it is one of those homeostatic mechanism which provides us or regulates the water balance. Now, next time I will talk about hypothalamic regulation of sleep. I have talked about sleep. Now I will concentrate on the hypothalamic regulation of sleep in my next class. Okay, so these are some of the references. So whatever I have taken some of those concepts because the recent concepts, they came from this physiological regulation of thirst and fluid intake from the News in Physiological Sciences 2004, and this article from Bichette in the Nutrition and Metabolism, Annals of Nutrition and Metabolism, appeared in 2018. These provide a very detailed analysis, because the older textbook may not have those detailed analysis what I had trying to project. Thank you.